So the digestive system, and hopefully this will go along well with your lecture for what you're doing there. So now digestive system functions, first of all, is ingestion. And then digestion can begin. And we could say secretion or excretion at the end, but we also have to talk about absorption. And then excretion or secretion. I like to say secretion when it comes to enzymes. I'm sorry, not enzymes, but hormones. Sorry, more endocrine. Secretion. When we talk about the kidney, we'll talk more about secretion as a form of excretion. But excretion basically is fecal waste being removed. So we'll go through all the different parts of ingestion and digestion. Now, of course, you have to know the difference between exocrine and endocrine. So most of your digestive system is exocrine, where your secretions, like enzymes, digestive enzymes, are going into ducts, not into the blood. So endocrine is secretions into the blood, right? So remember, blood hormones, right? And exocrine more into ducts. And there's different epithelial tissue that form these functions or produce these functions. So as far as ingestion, does anybody remember the macromolecules, the organic macromolecules? What are the four, but really three that we eat mostly? Anybody tell me? I'm sure you can. Like say we go to, we have a breakfast burrito. The actual tortilla, what is in that? And maybe the beans. What's the nutrient that's in that?
You're talking like carbohydrates, protein?
Yeah, carbohydrates, right? Carbs, carbohydrate. And you should know what a polysaccharide is. A polysaccharide is a long chain of monosaccharides. But what's the breakdown? What's the monomer? What's the monosaccharide that's really important in us, in mammals, and in plants too, really? Anybody know? Like we store this monosaccharide as glycogen, right? Plants will store it as starch. So we eat the starch, of course, but what is the monomer? Like if I want to hydrolyze a polysaccharide, what is going to be the outcome of that? The smallest particle. The smallest
Glucose?
Glucose. This is what we need to absorb, right? We talk about glucose in our blood glucose levels. And this is really important because our body is really designed to use glucose for glycolysis and then pyruvic acids to be put into the mitochondria to make ATP, right? We want to use this for our fuel to make our potential energy of ATP. Somebody said proteins. That's good. Proteins. So maybe we put some eggs in there with the egg whites, right? Proteins in our breakfast burrito. And what's the monomer of the proteins? If we hydrolyze, say, amino acids, we hydrolyze a protein or a polypeptide. You hear the word peptide, we have amino acids. All right, so there's 20 amino acids, but at least 8 of them are essential, meaning we have to get them from our diets. And of course, you know, amino acids and proteins are our structure. So we have the machinery in our cells, like ribosomes, to build proteins transcribed from our DNA to messenger RNA to build specific proteins. And you need all the amino acids to build all the proteins that our body needs. So protein's about structure. We don't want to use it for energy. We can if we need to. But this is about structure. What's another one? I'm sure there's some grease in there and in that burrito.
Fats, lipids.
Lipids. We got lipids, right? Lipids, especially triglycerides. And what do triglycerides break down? What are those neutral little tails, long chains? Some of them are saturated, some of them monosaturated, some of them are polyunsaturated.
Fats?
Fatty acids, right? Fatty acids. Fatty acids. You've heard of omega-3 and omega-6 fatty acids, depending on where that double bond is on that carbon, or which carbon has the double bond to make it unsaturated. So fatty acids are the breakdown. And we could also ingest nucleic acids, right? If we're eating animal products, especially easy to get nucleic acids, which break down to your nitrogenous base and nucleotides, base, sugar, and phosphate. So in the lab today, in your virtual lab, you could basically test for these breakdown monomers by using different reagents, like especially for glucose versus starch. You could use Benedict's solution to test for glucose, right? You could use different biurettes for amino acids. You could use iodine for starch. So you're going to see the different color changes in the test tubes in that virtual lab, right? So this is basically what we have to absorb. This is the form of the macromolecule that we could absorb, and mostly in our small intestine. So most of our digestion happens on the way down, from starting in the mouth, the oral cavity, through the stomach, into the small intestine, but most absorption happens in the small intestine of these monomers of the larger macromolecules. So we could start with the mouth or the oral cavity, and this is a very good histology slide. It shows you the different histologies of each salivary gland. So again, we're going to take, in your lab today in our APR, we're going to learn the teeth and what the teeth are made of, how that's a mechanical digestion. So ingestion, of course, the burrito comes in, right, from outside. But the digestion starts in the mouth, mechanically, with the chomping of our teeth, of course, and the chewing and the tongue moving the food around, the breakfast burrito around. And that's all mechanical. Even your cheeks get involved in mechanical digestion and breaking down of that food. And the food will basically be called a bolus once it all mixes together, because now there's some chemical digestion going on with your salivary glands, right? And there's 3 salivary glands, and they're slightly different. This is a large gland just anterior to the ear, right above the jaw, and that's called the parotid salivary gland, which is mostly serous fluid, really, which has a lot, serous fluid and serous cells, like alveolar serous cells, which are exocrine cells producing a serous fluid, have a lot of salivary enzymes. And most of that is about carbohydrate. So carbohydrate enzyme is called amylase. So salivary amylase, because you're going to hear amylase other places. So I just want to say this is salivary amylase. And salivary amylase will start to break down your starches. You know, the plant has stored carbohydrate as starch. So amylase will break down those polysaccharides, maybe to disaccharides, like 2-glucose, which could be called maltose. So salivary digestion, or carbohydrate digestion, starts in your mouth. So the parotid gland is mostly about serous fluid with amylase. And there is some lipase too, but that's also part of the serous fluid that starts to break down fats as well. So it's not just carbohydrate breakdown in the oral cavity, in the mouth. This is the submandibular salivary gland, which is a little bit more mucus. It has serous fluid. It has release of salivary enzymes, but it has mucus cells. So you're going to release a little more mucus. And you can see the mucus cells. And it kind of reminds you of goblet cells that were producing the mucin, the precursor to mucus. So look at the histology, the different cells. Now, acinar or alveolar cells, you've heard alveolar with the respiratory system, but most secretory exocrine cells are called acinar cells, and we'll see the difference when we get to the pancreas. So a lot of mucus from the submandibular glands, and these are on both sides. And then you have sublingual gland, which is mostly mucus. And sublingual gland, believe it or not, even though it's not as big as this parotid gland, secretes the most fluid into the saliva. So most of your saliva comes from your sublingual glands. And you can see the duct there going into the oral cavity, lined by that cuboidal epithelium, right, with lumens. And you can see the mucus cells, a lot of mucus producing cells in these sublingual glands. So take a look at this histology, and it will look at the animations in the MH Connect, and we'll look at the dissections, and we'll look at some histology. So we have a good idea what these differences are. So digestion begins in the mouth with some salivary amylase and then some lingual lipase as well, which starts to break down your lipids. So always go through these tables. I like these tables, especially before you or during your lab, or before you go into a lab exam, because it breaks things down for you nicely. You could see what the function is, right? You could see, the word origin, like acinar means grape. I think I just learned this like 2 years ago, that meant grape. Okay, so your myoepithelial cells, which kind of expand and constrict based on these epithelial local autoregulation when they're stimulated, right? So you have mucus cells and you have serous cells. So the serous cells are more about your amylase, salivary amylase in this case, because we're still in the mouth. So the burrito now forms a bolus and gets ready for its descending further down. And I'll go through all the parts. By the way, from your mouth to your anus is called the alimentary canal. So there's going to be accessory organs to that alimentary canal. So this breaks down the glands, the salivary glands in your mouth. Remember, they're paired. They're on both sides. So you see the percentages. You see 60 to 70% is submandibular. All right, there's a lot of fluid coming from the submandibulars. Right, and there's parotid less than the submandibular. So serous, mucus, and mixed. This is a general look at the lumens and the epithelium and the layers of your digestive system, like the different parts. And these generally go through all the way through, starting in the esophagus and going downward, like the pharynx as well. Below the oral cavity is the pharynx. So once we go into APR, we'll look at the different epithelial changes as you go from the oral cavity to the pharynx into the esophagus. So all along the alimentary canal, you have a mucosa, which basically lines the lumen. Here's the lumen. You can see that different ridged formation along the tube, let's say the esophagus. And then you have within the mucosa, you have an epithelial layer. Like when you learned epithelial tissue, you saw that epithelium lines all lumens, right? And that usually where your cells are that secrete things like goblet cells. It could be stratified squamous epithelium in the esophagus, non-keratinized, that you could have in the pharynx. You could have pseudo-stratified columnar ciliated epithelium. And then you have lamina propria, which is really important because that's a vascular layer. You have blood vessels, arterioles and venules, right, delivering oxygenated blood, taking the deoxygenated blood away. Then you have the muscularis mucosa. Now this is, of course, smooth muscle. So that's going to be different than the muscularis layer, which is outside the submucosa. And the submucosa is right below, or more external, I should say, to the mucosa, and more blood vessels, and you have your nerve plexus, because all tissue is, in one way or another, innervated via a nerve plexus, especially in the digestive system. Of course, a lot of it is autonomic, if you can remember from last semester. And there's glands, of course, and we'll talk about the different glands in different areas of the digestive system. And the muscularis generally has two layers, an inner circular layer. Now, this is not a sphincter. A sphincter is more of a valve that controls the amount of substance going in or out of an area. So an inner circular layer and then an outer longitudinal layer. You can see that by the way it's constructed there. Now in the stomach, there's 3 layers you're going to see of muscularis. It's the only place along the alimentary canal that has 3 layers. So the stomach has an oblique muscle layer as well. And remember, this is all smooth muscle, autonomic. There are some areas, though, that are voluntary skeletal muscle along the alimentary canal, but of course, mostly on top and the very, very bottom. And serosa is always the outer layer of anything, right, of an organ. And it's a different fluid, a more watery fluid, not mucus, right? Mucosa is coming more from the inside, right? You get more mucus into the lumens. Mesentery, you should always think of intestinal, gastrointestinal when you hear the term mesentery. And this is the connection, really anchoring tissue to other parts of the digestive system and also to other parts of the peritoneum or whatever cavity you're traversing with that digestive. Because the esophagus, of course, is above the diaphragm, and most of the gastrointestinal contents are below the diaphragm, starting with the stomach and the liver. So the esophagus is above the diaphragm. It's highly vascular and an extreme amount of lymph vessels. Now, lymph, if you remember from this semester, actually is about reabsorbing excess fluid. It's about carrying your dietary lipids, like from our breakfast burrito, the triglycerides eventually, or fatty acids. And also has some immune cells as well. So you think immunity, you think white blood cells when it comes to lymph. So these go together, especially here in the digestive system. Now we're in the stomach, and we're going to go through each part, but I just want to go through some of this histology just because this is probably the hardest part. So remember, the diaphragm is right above the stomach, and there's a hiatus, which is kind of like a hole opening of where the esophagus comes into the stomach. Now, sometimes the stomach contents could herniate up into that hiatus. And that's very painful. They call that a hiatal hernia. Sometimes it hurts a lot when you eat like raw vegetables and some roughage. And it doesn't have to be just the stomach. It could be part of the large intestine or small intestine that could actually herniate up into that hiatus. So very, very painful condition. Just to get an idea of that anatomy right there. So in lecture, you're probably learning a lot about this as well, the different cells, and we'll go through each cell that's important for lab. So again, mucosa, submucosa, and muscularis. Remember, the stomach has 3 layers of muscle. So you include that oblique layer. So take a look at that extra oblique layer, just internal to the circular layer and the outermost longitudinal layer. And you can see the columnar epithelium and lamina propria and extensive lymph system. And this is the anatomy. You look close at the gastric pits, and I'll show you what rugae is, and that's the ridges along the internal part of the stomach. And the gastric pits are lined with these cells and gastric glands. So know the histology here. Now in the stomach, and the esophagus is not going to absorb anything. The esophagus is basically a conduit to bring that bolus downward. And of course, you should be familiar with the term peristalsis, which again, starting in the mouth, you could chew, which is called mastication, right? Mechanically break down the food using your tongue and cheek to get the bolus down. And you could voluntarily swallow. Now, swallowing is called deglutition, right? Deglutition. And the first part of swallowing in the upper part of the esophagus is voluntary. But once it gets towards the lower esophagus, it becomes involuntary, autonomic, and circular contraction and rhythmic contraction of smooth muscle is called peristalsis. So even if we're eating our breakfast burrito and we decide to go on the jungle gym and hang upside down, that food will continue, that bolus will continue to go towards the stomach in that one-way direction thanks to peristalsis. So of course you need the autonomic nervous system to help that along and local regulation of these muscle cells. So, when you hear about things like neurological conditions like Alzheimer's or ALS, really horrible conditions, usually the problem in late stage Alzheimer's or ALS is they have dysphagia, which is difficulty swallowing, and they wind up aspirating the food into their trachea. Remember the epiglottis? You'll see this later in the lab. You'll see where the epiglottis is in relation to the esophagus. So the esophagus is posterior to the trachea, and the food has to be diverted into the esophagus. So if you have difficulty swallowing and autonomic conditions, nervous system conditions, and you have a hard time diverting that food through deglutition into the right tube, which is posterior, it can be aspirated into the trachea. And that can cause aspiration pneumonia. And a lot of people with those chronic neurological diseases will succumb to the aspiration pneumonia. And on the other hand, they have a problem with their diaphragm ventilating, of course. That's another cause of their death. So again, these are the gastric pits, and gastric, of course, means stomach. Now, this stomach is one area that your body's extremely acidic. So you can have a pH of 1.5 to 3.5 in the stomach, which is a hostile environment for proteins, right? Except you're going to learn about a protein enzyme called pepsin, which through evolution can survive in those highly acidic environments. And there's also a lot of hydrochloric acid, HCl, which basically has a pH of 0, which continually lowers the pH of the stomach. So the stomach is an area of protein digestion, breaking down protein. We're not absorbing a lot of things in the stomach, maybe some alcohol and some water and some dietary lipids. But it's not a great area of absorption, but a big area of protein digestion. So again, the stomach walls, you see, again, use these tables to go through each layer. And again, really the only difference here is the inner oblique layer. And I like what's saying twisting, right? Because I used to like say churning, which happens in the large intestine as well. But really converting that bolus into a substance called chyme. So now it's going to give you a creamy mass. Our breakfast burrito is now a creamy mass mixed with the coffee and the juice and everything. And always a serosa outside that alimentary canal. Here's a close-up of the gastric pits, and I'm sure you're learning about this in lecture. So you see the epithelial layer and the gastric pit with the lining of certain cells. There's mucus cells producing mucus, always mucus along the entire alimentary canal. And here's neck cell, mucus neck cell, which produces mucin, has the machinery to produce that mucin, which will be mucus ultimately in the lumen. Parietal cells are what's producing your HCl. Remember, HCl is basically a mix of your proton, your hydrogen ion, and your chloride ion. So this is your acid, right? And basically hydrogen is a proton, right? Its atomic number is 1. So if it loses an electron, basically it becomes a proton. So these protons are pumped into the lumen, and that's where the acid comes from. Okay, and again in the lecture you'll talk more about that. And also you have what's called intrinsic factor, which is released from the parietal cells. And intrinsic factor is important for absorbing vitamin B12. So B12 is very important. Now, what do we need? What's so big about vitamin B12? Well, vitamin B12 is very important to construct your red blood cells. So, and of course, you learned in the respiratory system, erythrocytes, red blood cells, are basically carrying oxygen on their hemoglobin molecules. So your oxygen carrying capacity is very important because you can't make ATP without the presence of a lot of oxygen, of course. So that's very important. So remember, parietal cells are about HCl, hydrochloric acid, and B12. Chief cells secreting a substance called pepsinogen, right? Pepsinogen. And pepsinogen, in the presence of HCl, will be converted to the enzyme that hydrolyzes protein, and that is pepsin. So most of your enzymes you're going to hear in the lab or have ASE at the end, but pepsin is one that has an IN, right? You'll hear about trypsin and pepsin, but think pepsin, stomach, pepsin, protein, peptide bonds, pepsin, right? And pepsinogen will be converted to pepsin via this highly acid environment that's formed from your HCl, which is released from the parietal cells. Now, enteroendocrine cells, now again, this is important for endocrine. Now, gastrin is really, I would say, one of the main hormones that's produced from your stomach, and it's a chemical messenger to the rest of the digestive system. So once these enteroendocrine cells find out about the breakfast burrito, they're going to send some information even lower to other parts of your digestive system as we go down to those areas. So some more histology here. The fundus is more superior. The pyloric region of the stomach is pretty much on the bottom. And you're going to see that in the lab. The top of the stomach, where the esophagus meets the stomach, is called the cardiac region. And the bottom of the stomach, where the stomach is going to go into the duodenum, the first part of the small intestine, is called the pyloric region. And there's valves or sphincters. There's your lower esophageal sphincter or valve, and then there's your pyloric sphincter or valve. And there's an upper esophageal sphincter or valve as well, which you'll see. Now, are we talking about the lower esophageal sphincter or the gastroesophageal sphincter of the esophagus, really? And there's a condition that you might have heard of called GERD. GERD. That's an acronym, right, for gastroesophageal reflux disease. So reflux is retrograde flow of, in this case, the chyme coming out of the stomach, refluxing into the esophagus. Now remember, the stomach is loaded with acid, and your esophagus is not designed to withstand high amounts of acid. So first of all, it's very painful, causing heartburn and burning in your esophagus. But you're destroying some of that stratified squamous epithelium, which is highly mitotic. So GERD not only is painful and discomforting, it also can lead to esophageal cancer if not treated. So basically, you need to stop the protons from being released. So you use a proton pump inhibitor as a medication, like Nexium, right? Will slow down that amount of acid that's going in there and treat it. It's not really, it might not be treating the actual reason you have GERD, and there's a lot of dietary things. And of course, it takes time to treat something like this. But at least get rid of the discomfort once you have this initial symptom by taking a proton pump inhibitor. So extremely acid within those gastric pits. And you can see the stomach lumen here. So get a good idea of the histology as you go through your lab. And these are the parts of the stomach, and the cardia is basically the top, and then you have the fundus and body, which we'll point out when we do the rudimentary anatomy of the digestive system. And the pylorus is down towards the bottom of the stomach. So we made our way from the oral cavity, right, through the pharynx, and then into the esophagus, and through the diaphragm into the cardia of the stomach, and then through the fundus and body to the pylorus of the stomach. And now we're ready to go into the small intestine. And the first part of the small intestine is a magical area, short but magical area, called the duodenum, or duodenum, whatever you want to call it, wherever you're from, right, Todd? So here are the cells of the stomach. Again, you probably do this in lecture, so this all should sound familiar if you're there. Your chief cells secreting that pepsinogen, right? So technically pepsinogen is called a zymogen, and it's converted to pepsin. So it's soluble until it's converted to pepsin, and pepsin, of course, is the enzyme, right? G cells, gastrin, those are the enteroendocrine cells, and you'll see that again in lecture, you'll see about enteroendocrine cells in the small intestine as well. So this is the major hormone, I like to say, of the gastrointestinal system. And the mucus cells, of course, they're called neck cells here in these gastric pits, and they produce mucin, which is basically the glycoprotein that's converted, and will actually convert to mucus, but the mucosa actually protects the stomach lining from this excess acid, right? So you need the mucus cells. You also need prostaglandin and other protective chemicals that help prevent the stomach from being eaten away from the acid. Now, you've heard of taking something like an NSAID, right? Non-steroidal anti-inflammatory drug. NSAIDs are like your ibuprofen, right? Which Advil, right? And Motrin, or your naproxen, which is like Aleve, and aspirin, right? Aspirin are non-steroidals, they're not steroids. That's acetylsalicylic acid. So these medications that we could take very easily because they're over the counter, right? But the problem is they stop the production of prostaglandin. They stop the enzyme that produces prostaglandin, which can decrease your protection of your stomach lining. And this makes a beautiful area for certain bacteria, like H. pylori, Helicobacter pylori, can form a nice little residence in your stomach. And the stomach eventually is going to be eaten away, and that's what an ulcer is, a gastric ulcer. So between the bacteria and the acid, you have this unprotected area that's going to ulcerate, and it's extremely painful, and you could actually lose blood cells from a situation like that. So again, the mucus is very important in the stomach. So, and again, be careful on how many NSAIDs people are taking or taking. My rule is always 5 days on an Advil, and then you might have to call your doctor and see if you need a different type of anti-inflammatory, depending on what you're taking the Advil for. Usually it's headaches, but not always, right? Back pain and so forth, so on. So another parietal cells as well, and that's what's producing the intrinsic factor and the hydrochloric acid. Our intrinsic factor is important for vitamin B12 absorption. In your surface mucus cells, of course, also causing a protection from the hydrochloric acid, which is a hostile environment. Amazing how something like pepsin survives that. So we're going down a little bit into the small intestine, which small intestine, of course, is the longest. The first part is the duodenum, and that's a really important part because a lot of secretions going into the duodenum. And then you have the jejunum, which is the middle part of the small intestine, and that is the longest part of the small intestine. And then the ileum. Look at the spelling. It's different than the ileum, the bone ileum. And notice where that is. This is important. It's in the right lower quadrant, the ileum, because that's the last part of the small intestine. So the first part of the large intestine, which is called the cecum, will begin in the lower right quadrant. So it's good to know where the beginning of the small intestine is, right? And where the end of the small intestine is. Very important. So the duodenum is going to be an area, of course, it has a lumen, but you're going to get secretions. First of all, the chyme from the stomach is coming into the duodenum, right? So that's still, that chyme is kind of acidic. You're going to get secretions from the pancreas. Now, the pancreas, of course, you know all about the endocrine function of the pancreas, but now we're talking exocrine, which the pancreas is mostly exocrine outside of the islets of Langerhans. So the acinar cells are producing enzymes, digestive enzymes, but not only that, the pancreas secretes bicarbonate, which is a buffer, a base that increases the pH of the chyme, because now we have to go down to the rest of the small intestine, which is absorption. And what does absorption mean? Absorption means we're bringing the contents, those macromolecule monomers into the blood. They're not staying in the lumen, they have to go into the blood, and our blood pH needs to be 7.4, right? So we also have bile coming in to the duodenum, and that's from the gallbladder and the liver. The liver makes the bile. The gallbladder stores and concentrates it. So a lot of fun stuff enters the lumen of the duodenum, which is also lined by a mucosa, has a submucosa, muscularis. And here's a term for the outer part, usually connective tissue, anchoring tissue, like in the blood vessels, it was called the tunica externa. But now we're talking about digestive tubes, so we could just call it adventitia, and it would mean the same thing. So along the jejunum, down here, now we have these cells, which are mostly simple columnar cells with microvilli and villi. Now, villi is kind of look like cilia, but cilia is for movement. A villi or microvilli is for absorption. That's for absorption, like little flexible straws, increasing surface area for absorption. What are we absorbing? We're trying to absorb those small macromolecules, your glucose, your amino acids, your fatty acids, right? So there's so many villi here that we basically call that a brush border. And you're going to have more enzymes in there, like brush border enzymes. Like you might get a disaccharide down in here, like if we had a little chocolate milk, right, with our breakfast burrito, there's a sugar in most dairy products called lactose. Lactose is a disaccharide of glucose and galactose, but we can't absorb lactose straight into this villi, the small intestine. We need to break that down to the monosaccharides. So lactase is the enzyme that will hydrolyze lactose into glucose and galactose, 6-carbon sugars. And now we could absorb it. But if you don't have lactase, it's not in your brush border genetically, or you have some type of intolerance to that particular dairy sugar, that's called lactose intolerance, right? And there's different reasons for it. But if you don't have the enzyme, you can't absorb it. And that's going to make you very uncomfortable. And this will continue to increase mucus, cause things like diarrhea, which is excess mucus, right? Excess fluid. So in the brush border, you have enzymes, and usually they end in ase, like amylase, right? Lipase and nuclease, right? Or protease, all those ASEs at the end, they're protein enzymes, right? So the pancreas deals with a lot of that. You're going to see again in the lab that the pancreas secretes, pancreatic amylase, pancreatic lipase. It also secretes a protein enzyme called trypsin. So remember, pepsin stomach, trypsin pancreas, right? And in lecture, you'll learn more about CCK, cholecystokinin, and its effect on bile release and other enzymes, and also hormones that are released. Okay, so pancreas, of course, adds a lot through the pancreatic duct to the duodenum, which really carries along into the jejunum. Now, the ileum is basically the last part of the small intestine leading to the large intestine or colon. And now you'll start to see what's called Peyer's patches. And this will bring in your lipids into the lymph system. So these are basically acceptors of lymph or absorbers of lymph, all right, for lymph system, which is dietary fats. And remember, in cardio, we talked about carriers for lipids like low-density lipoproteins and high-density lipoproteins. But lipids in the lymph system are carried by chylomicrons. Again, so if I have a little excess fat in that burrito, saturated fat, especially triglycerides, it will be absorbed here and brought into the Peyer's patches, mostly in the ileum. And that ultimately, the lymph is going to be reabsorbed into my bloodstream, into the veins via the thoracic duct up around the left subclavian, right? So those fats are ultimately going to get into my blood supply. As you know the story. When it goes into the veins, especially up in the left brachiocephalic vein, it's going to go into the superior vena cava and then into my right atrium. So ultimately, that blood is going to come back from the lungs and be pumped into my systemic circuit and also the coronary circuit. So you got to be careful on the saturated fats going through that area. So the last part of the small intestine is the ileum, and there's a valve between the first part of the large intestine, which is the cecum. Here's the ileum. It's called the ileocecal valve. Remember, a valve is for regulating the amount of substance that can be passed through to the next compartment. In this case, it's the cecum, and that's in the lower right quadrant. And you'll see that in the lab when you do the rudimentary anatomy of the alimentary canal. You can see it's in the lower right quadrant. And you'll see this appendix, which is worm-like. That's why they call it veriform. Now, the appendix, I'm not sure exactly what it's doing as of now, but whatever this is, copyright 2019 probably, it's lymphoid tissue, it's immune cells, but it also could be an area where our normal symbiotic microbiota, good bacteria, are housed and like to proliferate, and also which add good things, which add positive things. They help us absorb vitamins in our large intestine. Right? They help the absorption of vitamins and the construction of your flora, your intestinal flora. So, of course, if it's inflamed, it's going to be very painful, very dangerous, because if this ruptures, you're going to get what's called peritonitis, a severe infection in your peritoneum, and this is a surgical emergency. So we really don't know much about the appendix until something happens, right now. So but this is where it is, and anatomy, it's right off of the sacrum in the right lower quadrant of the abdomen. Then you have the ascending colon, which moving now we're getting more into fecal material, because most of what we need to absorb has been absorbed. So in the large intestine, you're basically forming waste material, fecal material, and absorbing some water. And of course, the vitamins are important here in the large intestine, and the bacterial flora really help that absorption of vitamins. So the ascending colon goes upward to the right. Then you're going to have a flexure here, which is called the hepatic flexure or right colic flexure, because it's going to make a hairpin turn across the mid-peritoneum. And this is larger, of course. Now, we're just looking at the large intestine. You're going to remember the small intestine is covered here. So, when we do a fetal pig dissection, you can see how that all is lined up. It's very vague looking, and it was the same color, but the size is bigger. It's shorter than the large intestine, small intestine. And you can see the transverse colon pretty plainly on the models in your atlas. It's kind of easy to see the transverse colon, but you also got to be careful looking to see what's been dissected. Like, you have to look superficial to deep. And obviously in this cartoon right here, you can see that the small intestine has been dissected out. So you have to be really careful of that. Now the spleen is on the left side, so this hairpin turn is called the splenic flexure. Like, this is the hepatic flexure. And this is the splenic flexure of the colon, also known as the large intestine. And the spleen is here. Sometimes this is called the left colic flexure. And then the fecal material and water goes downward on the left side, and that's called the descending colon. And then it kind of forms like a little S, and the last part of the colon is the sigmoid colon. And technically right here, this is called the sigmoid flexure. And then the last part before the anus, it's not really, technically it's not part of the colon, it's the rectum. And the rectum is basically, nothing's going to be reabsorbed there. The rectum is basically a garage for the feces before it goes through the anal sphincters and out the anus. Okay, so again, mucosa, submucosa, muscularis with 2 layers everywhere except for the stomach, 2 layers, circular inner, longitudinal along the long axis of these conduits, these tubes. It's a layer of muscularis. And a lot of mucus here, goblet cells, in your glands, of course, in blood vessels, in the lamina propria. Now, again, this is a little different. It's these little outpocketings, or I should call these little segments of the whole colon. You can see them there, called haustra. And you have this haustral churning, again, with the release of gastrin causes that smooth muscle contraction. And that moves very big bulk flow, a bulk movement, like peristalsis, but we call it haustral churning. And there's a ribbon-like muscle too that goes along the long axis, outer part of the colon, and it's actually a ribbon-like muscle. It's called the tenia coli. Sometimes I put an A there, tenia coli. And you'll see that in the lab, you'll see what that is. And always check the checklist that I put on Blackboard so you see what you're responsible for, and of course all the dissection and animations on the MH Connect. Now the liver, and again the MH Connect will go into the liver. It's complicated, of course, and you're doing this in lecture. So the liver here is in mostly the upper right quadrant, but kind of comes across the middle. So again, when you look at your atlas, you look at your APR, you see that the stomach is in front of the pancreas. The pancreas kind of goes to the middle of the abdomen, and the stomach is more upper left quadrant, and the liver is mostly in the right upper quadrant. And you can see that little green organ right there, which is the gallbladder. So of course, the cells of the liver are called hepatocytes. Anything hepatic is liver. Sometimes the gallbladder, you use the word cystic, like here's cystic duct, and that has to do with the gallbladder. Hepatic duct has to do with the liver, of course. Now, the blood vessels in the liver are very porous. So the blood is basically percolating through here because the liver is a massive amount of detoxifying, a lot of peroxisome in these cells. And it's a massive area of manufacturing hormones and enzymes, and conversions, and breaking down red blood cells into their parts. A lot goes on, making bile. There's so many things that the liver does that you're learning in lecture. So get to know some of the parts, of course. So the thing about the liver is it's receiving blood from the intestines, because remember all those things that we're absorbing, all of that goes back to the liver via a portal vein, which you're going to learn about. So the portal system is usually a blood system within an organ system. If you remember the hypophyseal, hypothalamic portal system of the pituitary gland. But this is all those nutrients coming back to the liver to be, like, for example, all the glucose coming to the liver to be converted to glycogen, right? Glycogenesis, building glycogen, because we store carbohydrate in our liver. So glycogenesis is a big production within the liver, and done by the hepatocytes, which is really important. It's under the control of other hormones when you did endocrine. So this liver is, of course, a great accessory organ to digestion, but also important for the endocrine system and other systems. So again, making bile. So again, go through the basic parts, the sinusoids, the hepatic portal vein, which you'll see the central vein. So all the blood from the liver will be sent back to the inferior vena cava via the hepatic vein, or hepatic vein proper. So that, of course, is a vein returning blood. Veins drain back to the heart. So, and then there's hepatic artery also, which brings oxygenated blood to feed the parenchyma, the functional cells, hepatocytes, oxygenated blood, so they can undergo their function. And there's, of course, there'll be a list coming up about all the lobes, the quadrate and caudate, and the falciform ligament that separates these two lobes here. And we'll see that in APR. You'll see the different anatomy parts, and use your checklist to see all those main parts. So here's, again, the lobules in the hepatocyte, and the triad, portal triad, central vein, which you saw in the picture before. And you can see the histology here with the central vein always in the middle there, and all the surrounding hepatocytes. Pancreas, again, here's the duodenum and the main pancreatic duct. Very plain to see in real anatomy. You could actually see that thickening. Visual gross anatomy, you can see where it's going into the duodenum via the accessory portion as well. Ampulla is always like an area of dilation within a duct or vessel or organ too. And here's the second part of the small intestine, the longest part, the jejunum, right? The jejunum. So again, the pancreas has endocrine function in the islets of Langerhans, the alpha and beta cells, right? The beta cells producing insulin, the alpha cells producing glucagon, but most of it is acinar cells. Acinar cells producing exocrine hormones like trypsin, pancreatic lipase, pancreatic amylase, and also that bicarbonate fluid that's secreted into this lumen of the pancreatic duct, which ultimately goes into the duodenum. And you can also see here, it's showing the common bile duct, which is bile coming from both the gallbladder and the liver. So here's the histology of the pancreas, which you probably saw a bit of it when you do endocrine, of course, in last semester. So this is a nice view just to go through, and you'll do this in APR, the oral cavity, right? And this big muscle right here is the tongue, right? So you can go through this and do this. You can see the parotid gland here, giving that serous fluid with the mostly salivary amylase, submandibular salivary gland, which secretes most of your saliva from that, sublingual. And then you see the beginning of the esophagus, which is here. But don't forget, there's a pharynx, a nasopharynx, an oropharynx, and the laryngopharynx. That really, and especially in the oropharynx and the laryngopharynx, the two most inferior parts of the pharynx, that's where the food and the air are mixing, right? So that has to be, the food has to be diverted posterior into the esophagus. So here's your esophagus, number 6 here. And don't forget where the diaphragm is. I know you won't because you did respiratory and you did muscle system last semester. And here's the liver, and you can see that kind of comes across. You can't really see the pancreas too well, but here it is. So the stomach is in front. So the first part of the small intestine is the duodenum. And it's hard to say, but the middle part is going to be the jejunum and the ileum, the last part. So you just got to be careful where we put the X's. And here's the veriform appendix with the cecum, ascending colon, transverse colon, descending colon, sigmoid colon, and then the rectum, and then the anal canal right there. So that's basically the trip from the oral cavity, right, and the parts that we went through. So we'll go through the teeth in APR, all the different names, the deciduous and the adult teeth, and what teeth are made of course, the enamel, dentin, ligaments, and you'll see that in APR. Very straightforward. We have everything in the checklist that you need to go over. So the regions of the stomach, the body, the cardia, the fundus and pylorus, and more features of the stomach, the gastric folds, rugae, not like Bob Marley reggae, that's rugae, right? Those, it's kind of like gyrus in the brain, except it's inside the lumen of the stomach, the greater curvature. You'll see the greater omentum, that's on your list. The greater omentum is like a curtain of fat, or a fold of fat that comes across your abdominal area, right? Like an apron, really. And it's mostly triglyceride. And did you ever notice that your mediastinal and your thoracic cavities has well protected or structures like the sternum. It has the sternum to protect the heart and the ribs to protect the lung areas. And down in the pelvis, you have your os coxa and your ilium, pubic bone, and all that's protecting. But in the abdominal area, there's no bones. It's just straight up organs, right? So you have the greater omentum, of course, which has more function than just protection. There's insulation, and it also, the triglycerides are secreting hormones like leptin, right? Is secreted from the triglycerides or from the adipose tissue that's in the greater omentum. And there's a lesser omentum too. And you will have to go through APR to show you where the lesser omentum is structurally, but just get used to these terms. And the cardiac sphincter is the lower esophageal sphincter, also known as the gastroesophageal sphincter. We talked about GERD, right? That was important. That's basically a valve that controls that bolus coming into the stomach, the cardiac part of the stomach, which is the beginning of the top of the stomach, we should say, most superior, right? And then down the end, and of course, lesser curvature and greater curvature are easy to see once you do the lab. Pyloric sphincter. Now, pyloric sphincter is the bottom of the stomach or the end of the stomach, and that is a valve that controls the chyme going into the duodenum. So it's really important that you get that flow through the alimentary canal. So again, like the lesser curvature, simply this here, and the greater curvature is here. Cardiac notch, right? You've heard of that term before, right? So here's the duodenum here. So the pylorus is the end of the stomach, basically. The cardiac region is here. Here's the esophagus, and now you can see the rugae, which again, in the lab, you'll go through each one of these in the anatomy and physiology reveal, and they highlight it for you. The models are pretty good too, but you know that I like the anatomy and physiology reveal dissection the best for us. And if you noticed on the exams, that's what I like to use the most, because the atlases are great, but sometimes they over highlight things and they take away from what you're really looking at. But they will be used as well. So go through all that. And this is good too when you're going through your liver and looking at all the different parts of the liver, the lobe and the porta hepatis, quadrate and caudate, part of the right lobe, falciform ligament, of course, and then the bile duct, common bile duct, and common hepatic duct. So these two actually are forming the common bile duct. So the bile is coming from the liver and the gallbladder. So the gallbladder is not producing the bile. The liver is making the bile. So the gallbladder is storing and concentrating it, right? The gallbladder. So that's kind of easy to see in the right upper quadrant, the gallbladder. So remember this too. The bile is not an enzyme. Bile is an emulsifier for lipids, right? So that's important for making them a little bit more hydrophilic than they are, right? So because it's basically like a detergent. Like say, I'm eating my breakfast burrito, and of course I spill everything on my shirt. Like they got a grease stain, which is lipid, and they got some protein stain from the beans and rice, and from maybe I had a little egg in there. So of course proteins are hydrophilic, and fats, grease, is hydrophobic. So again, going down into the intestinal system, you have to kind of make it more hydrophobic to be broken down by the enzymes. So the detergent will actually make my stains amphipathic, which is both hydrophilic and hydrophobic, polar and non-polar. So it could be broken down in the laundry. From what I've heard, I've never seen that before. My wife does that. So like I have dirty clothes, they come back clean. I don't know how that works. So this bile basically changes the chemistry of the fats by emulsifying, maybe making them a little bit more hydrophilic or amphipathic, so they can be broken down easier the rest of the way down the intestinal tract and then reabsorbed and absorbed better. Right. So it's about fat metabolism, basically, or fat digestion, I should say. Now, do we need a gallbladder? I mean, we don't need it, basically, because we have the liver producing and secreting the bile into the duodenum, but you'd probably miss it, and you probably have a harder time, going to Popeyes and eating your dietary fats if that happens. Okay, so we'll go through all that. And the pancreatic duct, easy to see. Duodenal papilla, take note of that. It's probably not on our list, but make sure you know where all the organs are. And the pancreas has three parts: the body, the head, and the tail. because it looks like a fish, like a fillet of sole, I think they used to say. So again, go through this and use you can use this if you want. I mean, you have the practice in MH Connect, which is great, and APR and Atlas, where you could practice. But here's the gallbladder. And again, going into, and of course the kidney's involved here. Next week, we'll see the kidney. We'll talk about the adrenal gland. Spleen is always on the left. You can clearly see that pancreatic duct because we're looking at the pancreas now. The tail of the pancreas over here and the head here. So this is the duodenum. The stomach would have been here. So remember, that's been dissected out. And you already studied the vasculature, the arteries, the celiac trunk, right, coming off the abdominal aorta. So you have a good idea of where all this is as you're doing your lab. This is an inferior view of the liver. You can see the central vein. And again, the gallbladder. And you'll find the portal veins and all that. We'll go through that. This is real anatomy, showing these mesenteric vessels. And again, circular folds in the jejunum and ileum, slightly different, right? There's more absorption in the jejunum there is in the ileum overall. Vasa recta, of course, is blood vessels. We'll see a lot of that in urinary next week, what a vasa recta is, and nephron. But you can see here that it's vasculature, mesenteric vasculature, controlled extensively by the autonomic nervous system, but there's also what's called an enteric nervous system. It's so extensive in the gastrointestinal system that it's autonomic, but it's part of it. It has its own autonomic system called the enteric, which you could see. So again, more of these areas that you go through as you're studying and doing the lab. Haustra, I mentioned, Taenia coli, and the epiploic appendages you'll see on the greater omentum. This is very simple, like the atlas will show you. Make sure you know where the diaphragm is, even if it doesn't tell you there. And you can see the esophagus coming through that hiatus, and the liver and its falciform ligaments, and the stomach. And the pancreas can't really be seen too well here. Transverse colon. So it looks different on an atlas model, plastic model. Descending to sigmoid colon, and the ascending colon here, cecum, down there. And this would be where we point to the ileum, I-L-E-U-M, just to get a look. And then you can see the pancreas now because the stomach has been deflected out. And you can see the spleen. And the duodenum gives you other areas, and the right colic flexure or hepatic flexure right up here. And again, you can't see the small intestine in this case. You can see the mesentery, the connection in the blood vessels. And here's the rectum and the cecum. And this would be your ileocecal valve. Just some highlights as you're going through. And there's a list. And we can do that together before you go into it. Here's a real anatomy extracted colon, right? Which shows the cecum. Here's the appendix. And ascending, remember, everything's going this direction, up to the hepatic flexure, across mid-peritoneum to the splenic flexure, and then downward to the sigmoid colon, to the rectum, to the anus. Right? And this Taenia coli is that ribbon-like muscle. And it's hard to see, but each portion is called haustra. Again, we're not absorbing much here, just water mostly, some vitamins. And this is where intestinal flora is mostly, or microbiota, normal bacteria. Gallbladder, right? Showing a stone, gallstone, because this does cause a lot of problems as far as inflammation. They call that cholecystitis, which is inflammation of the gallbladder. And cholelithiasis is gallstones. So that's bile, highly concentrated into crystals, then it forms a stone, which can obstruct that cystic duct and cause a lot of pain, especially if you're eating fats and you have Popeyes in there. A lot of saturated fats or triglycerides are present, and this starts to get more release of bile, and all of a sudden it just kind of gets bigger and more painful. and can cause a problem. Can cause a problem. And, or you could take the gallbladder out, you can remove the gallbladder and still be okay. I mean, would you miss it? Yes, especially if you're eating fats. So again, like we started this, you have to know your macromolecules and the subunits of each, because that's what, when you do the virtual lab, you'll have a look at what you're testing for and what different reagents you use to see what is present, especially if you add an enzyme to it, like an amylase to a starch, right? Or pepsin to a polypeptide or protein, or lipase to something like buttermilk, right? Because it's going to break down or hydrolyze to fatty acids. And sometimes you have to check the pH, right? You use litmus milk to see if the pH goes up, which means there's a higher presence of the fatty acids. So your virtual lab is really cool. So go through that on your own. I'll stay with you if you need me to go through that. Okay, and here's the alimentary canal and where things are digested, right? Like we talked about before, all the way through. So small intestine is really where all the fun happens, where these nutrients are being absorbed most. So remember, ingestion, digestion, which is mechanical and chemical. Chemical is your enzymes. Then you have absorption, which happens mostly down here in the brush border of the small intestine. So here's all your macromolecules. And like I said, nucleic acids is another macromolecule. We eat the nucleic acids of animals especially easily. So you don't have to eat animal products to get all these, of course. Of course, this is not a nutrition class, but you do have to know that as long as you're getting your essential amino acids, you could eat plant-based all for the rest of your life and not have to worry about proteins, the recommended daily allowance or daily amount that you need for each of these nutrients, and vitamins as well. Okay, so this will be part of your chemical digestion. You use the Benedicts for your monosaccharides, and you use, I believe, iodine for starch in the experiment. And this is basically your colon. So that's the end of the colon, rectum,